Hello everyone. So in my last video I had discussed about the classification of non-chordates. So today we will move on with the chordate classification, and the last part of this topic will be some portion of developmental biology. So just giving a quick recap about what we had discussed in the last video. So this animal kingdom, that is a multicellular kingdom, and it consists of two different levels of organization. One is the cellular level and the other is the higher level, which is tissue or organ level. Now when it is in the cellular level, we have one phylum which is Porifera, and in tissue or organ level, according to symmetry, there are two types. One is radial and the other is bilateral. In radial, as I had already discussed, that Ctenophora shows biradial type of symmetry, and radial symmetry is shown by Cnidaria. But the poriferans are asymmetrical, and in bilateral symmetry we have the acoelomates without any coelom, Platyhelminthes, the pseudocoelomates or false coelom, the Aschelminthes, and the true coelom or coelomates are annelids, arthropods, mollusks, echinoderms. So in the last video I had stopped till echinoderms. So today we will start with hemichordata, and then we will move on to the chordate classification. So for that, giving a quick recap, Porifera are the members known as sponges, and they have pores in their body which are called ostia, and they act like channels. So water enters through these pores and comes out through the large pore called osculum. And these sponges, they have a body cavity which is known as spongocyl, and this spongo cavity is lined by cells called choanocytes. So an example is cycon. And Ctenophora, moving on to the next phylum, they are known as comb jellies, and the comb plates, they help in locomotion. Like I have already discussed, that 8 comb plates are usually there in the Ctenophorans, and they have a special property known as bioluminescence, or they emit light. They are also marine organisms, and before that I forgot about Cnidaria. So in Cnidaria we have two forms, polyp and medusa. I have already said that polyp is the stationary form and medusa is the free floating form of a jellyfish example, and they are known for their defense mechanism. Like we all know how jellyfishes can sting, so they have different kinds of mechanism for capturing prey and anchorage. And moving on to the next phylum, which is Platyhelminthes, we know they are known as flatworms. So their body is very flat and it's like a ribbon. So example is a tapeworm, liverfluke, etc. And Aschelminthes are the roundworms. So moving on to the next phylum is the Annelida, where the body is segmented, which is known as metameric segmentation, which is the repetition of units along the length of the body. And then comes the arthropod, the largest phylum, where we have all the insects, all the crabs, all the prawns, etc. And they have special body which is divided into head, thorax, abdomen, and they have jointed appendages, which is their characteristic feature, as well as some organisms, they have different respiratory organs like gills, book gills, tracheal system, etc. Moving on with mollusks, I had already discussed that they have a soft body, and they have a muscular foot, and also they are sometimes covered by a shell and sometimes not, like octopus and snails, etc. Echinoderms, I have already said, they are spiny skinned organisms, and they have a special kind of system called water vascular system, which helps them through their excretion, through their nutrition, and etc. Example is the starfish. So like I was saying, that what are exactly chordates and what is different from chordates and between chordates and non-chordates. So the first thing is the notochord. Presence of notochord is what defines chordate. So notochord is nothing but a cord-like structure. I will be coming to that later. Is at the dorsal portion there is this cord like thing which is known as notochord, and above that there is this dorsal hollow nerve cord. So presence of these two are mandatory for being called a chordate. And whenever there are presence of pharyngeal gill slits, at least in one stage of the life cycle, we can call it a chordate. The heart is ventral, post-anal tail is present, that is above, beyond the anus, there should be a tail region. So at any part of the life cycle, at least they can show it throughout their life, or at any part of the life cycle it must be present for being called a chordate. So these are the four basic features of chordates: notochord, dorsal hollow nerve cord, pharyngeal gill slits, and post-anal tail. These are the four characteristic features of chordate. And due to the absence of all these features, we can call the rest of the organisms as non-chordates. So now a very interesting phylum is there, which is known as Hemichordata. Like from the name itself, you can understand that it is like semi or half chordate. So why it is called like this? Why it's called Hemichordata? So they have this kind of a structure. They have a proboscis in the anterior portion. They have a collar and a trunk. Their body is divided into like this. Three parts: proboscis, collar, and trunk. There are gill slits for respiration, but there is no post-anal tail, and also there is a rudimentary notochord, which means it is almost on the verge of getting deleted. But this rudimentary notochord, the presence of it has given its name as half chordate or hemichordate. But now this theory has been demolished, and molecular studies have shown that hemichordates are more closely related to echinoderms, the previous phylum, rather than the chordates. So therefore there has been studies showing that hemichordates are related to echinoderms much better and in respect to chordates. So therefore they cannot be called as chordates anymore, or but their name is still there, and therefore they are separated from phylum Chordata, and now they are as a separate phylum as Hemichordata before Chordata, and they are commonly known as acorn worms. And also there are two classes to it, Heteroneusta and Pterobranchia. So these two classes are there. There are a few examples given here, and this is the basic structure of a balanoglossus, which is a very good example of class phylum Hemichordata, and these are the structures of the acorn worms. So just before the non-chordates are ending and the chordates are beginning, so there is this phylum called Hemichordata, and after that we will move on to phylum Chordata. So phylum Chordata, like you can understand that this phylum has all those characters that I was talking about. It has a dorsal hollow nerve cord. In non-chordates, the nerve cord is there, but it is usually ventral. So in cockroaches and all that, we usually dissect from the dorsal side because the nerve cord is in the ventral side. And usually the cockroaches are going like this, and the nerve cord is on this side. This is the ventral side, not on the dorsal side. But in our case, it is on the back side. So we have a dorsal nerve cord. This is a basic feature of a chordate. And there is this notochord, which usually gets transformed into a vertebral column in case of vertebrates like us. And then they have this pharyngeal gill slits, which we do not have as an adult, but it is the characteristic feature, and at least in one stage of the life cycle, even in larva or in embryo condition, one should possess these characters, all these characters. And the anal opening is there, and beyond that there is this post anal tail, which is also a very important feature of phylum Chordata. So this phylum has been divided broadly into two parts, protochordates and euchordates. Protochordates, as you can understand from the name, these are primitive chordates, lower chordates, and these are acraniata, which means they lack a skull or a brain box, as we can call it. They lack this cranium. But in euchordates, there is the presence of cranium, and they are known as higher chordates. So these protochordates, they are divided into two subphyla, Urochordata and Cephalochordata. And these euchordates are divided into the one big subphyla, which is known as Vertebrata. This Vertebrata can be divided into two superclasses, Agnatha, Gnathostomata, presence of on the basis of presence or absence of jaws. So the jawed animals are categorized into gnathostomata, and the gnathos meaning jaw, stoma means mouth, so they wear jaw in their mouth, and agnatha means they lack jaw. So the one and only class of this division is the class Cyclostomata, and in the in Gnathostomata there is two superclasses, Pisces and Tetrapod. Pisces are obviously the water animals or the aquatic animals, which are mostly fishes. There are two classes, Chondrichthyes and Osteichthyes. Chondrichthyes being the cartilaginous fish and Osteichthyes the bony fishes. And we have in tetrapods, the superclass tetrapod, we have the classes Amphibia, Aves, Reptilia and mammals. So this is the basic classification of phylum Chordata. Now coming to Urochordata, which is one of the subphylum of this Protochordates, we have Urochordata. So Uros meaning tail and chorda comes from notochord obviously. So the presence of their notochord in the tail region is what defines them as a chordate, and therefore the name comes from there as Urochordata. So they are also called tunicates because they have a tunica or the covering, outer covering, and this is known as tunic or the outer covering, like you can see, and this is the notochord which is there in the tail region of the larva. So therefore the name comes like this, Urochordata. So these are a few examples given here, and the adult loses the notochord, but since the larva contains this notochord, we can call it a chordate but a protochordate because it has primitive characters. Next is the cephalochordate. So like the name suggests that the cephalochordates, they have the notochord present from the head to the tail like this. The notochord is there continuous from the head to the tail. Cephalo means head, chord comes from notochord. So this phyla has the organisms which are known as lancelets. And also they are acraniata. Like I've already said, they lack a particular bony skull box. So therefore they are acraniata, and they are also cephalochordates. They are also protochordates because they have primitive features, and all these cephalochordates, they are called lancelets. A very good example is the Amphioxus, and Amphioxus, myxine or hagfish. These are very common examples of cephalochordates. So their basic feature is that notochord extends from the tip of the tail to well anterior of the body, and they do not have a brain case or cranium, and therefore they are also called acraniates. And they have a mouth, but they do not have any digestive system, and so they are quite primitive in nature. Next comes the superclass Agnatha. So in euchordates or in the true chordates, the first superclass that is there is the Agnatha superclass, which means they lack jaws. So they do not have jaws in their mouth, and the one and only class of superclass Agnatha is class Cyclostomata. So in class Cyclostomata, I'm sorry, I said that cephalochordates example was myxine, but it's actually Cyclostomata. I got confused. So this class has this members, they have suckers in their mouth. So the mouth is circular, and as you can see, there is no jaw like we have jaw. That they do not have jaw, but they have suckers around their circular mouth, and they are adapted so that they can adhere to any rock or any other fish, and as ectoparasites they can draw nutrition from there. So they have this capability of drawing nutrition from there, and they have 6 to 15 pairs of gill slits. So here, if you can see, there are gill slits present on their body, which is also a very good character of chordate, and they are actually ectoparasites, and therefore they do not need to have a proper digestive system. So they have a mouth with without jaws, and it is capable of sucking, so they can suck the nutrition from whatever fishes they have adhered on. So they have this capability. Next, coming to the to the other superclass, that is Gnathostomata. So superclass Gnathostomata has all these classes. First, if we start with amphibia, let us start with fish first, then it will be a perfect order. So fish, obviously, they have breathed through the gills, they have scales on their body, they have fins which help them to move underwater. They are cold-blooded animals and they are born from eggs. Coming to amphibia, they have a thin skin which is a moist skin. They can breathe through the skin, and also they are born from eggs. They are cold-blooded and they can live both on ground and on water. Next comes the reptiles, which are cold-blooded animals, but their body is covered with scales. They cannot, they live on the ground or in water. They are also born from eggs. Then comes the birds, whose bodies are covered with feathers. They usually have the porous bones, which are air-filled bones, so that it helps them to be lighter and it allows them to fly. Their mouths are toothless, but they have beaks, and therefore they are warm-blooded animals and they are born from eggs. Next comes the mammals, which is different from the others because they give birth to their babies and they not by eggs. Very few exceptions are there which are egg-laying mammals, but other than that they mostly give birth to their babies and they have hair throughout their body. They can breathe through lungs and they are warm-blooded animals. So from this we can move into the classification of superclass Pisces, which is Chondrichthyes and Osteichthyes. So Chondrichthyes means that their skeleton is made up of cartilage like sharks, and Osteichthyes meaning that they are bony fishes and their skeleton is made up of bones. So the body sizes, obviously that sharks are quite larger in size than the normal fishes, and the mouth position of the sharks is ventral, and whereas this is terminal, like at the end point the mouth is there, but for the sharks it is in the ventral surface. And their scales are different. Sharks have placoid scales, chondrichthyes, and the bony fishes, they have cycloid or ctenoid scales. Number of gill slits are also different. Here it is only 4 pairs, and it is in bony fishes it is only 4 pairs and it is covered by a bony operculum. But in cartilaginous fishes there are 5 to 7 pairs of gill slits. And if you look at the caudal fin, it is heterocercal, which means that the upper lobe is longer than the lower lobe. It is quite unequal in shape, so it is known as heterocercal. But here it is called a homocercal because both upper lobe and the lower lobe are equal in length. So these are some common differences of chondrichthyes and osteichthyes, or cartilaginous fishes and bony fishes, and therefore they are different in a lot of aspects. So next, moving on to the next class, which is class Mammalia. So class Mammalia, we have these subclasses which are Prototheria, Metatheria and Eutheria. So Prototheria means obviously the primitive mammals, which are egg-laying mammals, and they are also known as commonly monotremes. So, and the next is examples are duck-billed platypus and the anteaters. Next comes the Metatheria, where it is a common example is marsupial or kangaroos. They give birth to their young ones, but the young babies stay inside their mother's pouch until they mature. And then comes the Eutheria, or the placental mammals. They give birth to alive young ones and they do not need to be mature, but they can give birth to immature young ones as well. So from here, if we just give a recap of what we did, so phylum Chordata comes under 3 subphylum mainly, which is Urochordates. Cephalochordates are the protochordates, and under Euchordates we have vertebrata. So these are also called tunicates and these are called as lanceolates. Next, coming to this division, where we can see that vertebrata is divided into 2 superclasses, which is the divisions, which is Agnatha and Gnathostomata according to the presence or absence of jaws. Agnatha has only one class, which is Cyclostomata, and Gnathostomata has two superclasses, which is Pisces, which bears fins and they are water animals, and Tetrapods, which bear limbs and they are land animals. So amphibia, reptiles and mammals. And next, moving on to phylum Chordata, I have discussed what are the main characteristic features: the pharyngeal gill slits, the dorsal hollow nerve cord, and the notochord. So according to this, if we just compare the what are hemichordates, urochordates, cephalochordates, the prime thing is that these hemichordates, they have a proboscis and a rudimentary notochord. The urochordates, they have a notochord only in the tail region of the larva, and the cephalochordates, they have notochord throughout, from the tip of the tail to the anterior head, throughout the length of the body. So there are some common non-chordate features also, like the hemichordates, they have this dorsal blood vessel, and urochordates, they do not have any nerve cord. So that's why we can say that although they are classified in under chordates, but they are protochordates, because they are not considered as true chordates but primitive chordates. Next comes the subphylum Vertebrata, where I have already discussed that what are the basic features of these classes. From class Pisces to class Mammalia, there are certain changes, and in class Mammalia we have this subclass Prototheria and another one Theria, and infraclass Metatheria and Eutheria under the subclass Theria. So like I was saying that the monotremes are the egg-laying mammals falling under subclass Prototheria, and the Metatheria, there are marsupial mammals where like kangaroos and koalas, opossums and etc. And then we have the subclass Eutheria, or the placental mammals, which are viviparous and they give birth to their babies directly without any egg formation. The egg is formed, but without any hatching of eggs. There is direct placenta to mother connection, and then the baby is born. So coming to these terms, what are exactly oviparous, what is ovoviviparous, and what is viviparous? So viviparous means that they give birth to live young ones directly, and it's nourished by placenta like the placental mammals or humans. Oviparous means the egg-laying animals, or the ones which lay eggs, and outside the mother's body it hatches and the baby comes out. The third one is a very interesting one, which is ovoviviparous, and that means it's egg live birth. The eggs are already fertilized inside the mother's body, and the fertilized eggs are laid by the mother, and therefore the egg yolk provides the nourishment to the young ones, and then after a short time they hatch and they come out, or the eggs hatch inside the mother and the young are born live. Any two can happen, anyone can happen, and therefore it is a common example in case of some sharks, some reptiles, some snakes, etc. So moving on to the part where it's a little bit of developmental biology involved. So what are gametes? So gametes are basically the egg and the sperm, the female reproductive unit egg and the male reproductive unit sperm, and when they fuse together it forms a zygote. It forms the diploid cell, the first diploid cell of an organism, and it is called a zygote, 2N. So as a result of this fertilization, a zygote is formed, but there are different types of gametic fusion. First type is the isogamy, which is a quite primitive form, but here the two gametes are equal size. They may or may not be mobile, but they are of equal size. Therefore, it is called as isogamy. In anisogamy, one gamete is small, usually the male one, and one gamete is quite large, usually the female one, and they both can be motile or non-motile. But in case of oogamy, which happens in case of humans, or in other higher organisms as well, we can see that the male gamete is very small compared to the large female gamete, and the male gamete is motile because it has a flagella or tail like thing, and the non-motile is the female gamete, therefore it does not have any flagella or anything. It will not move. It is non-motile and it will stay at one position, and the male gamete will will migrate towards the female gamete. So these are the three basic types of gametic fusion, and from here if I just move on to cleavage. So cleavage is the series of cell divisions that occur to form a multicellular organism from a single diploid cell, which is the zygote. So the zygote first breaks down, the fertilized egg is the zygote actually. So it first breaks down into 2-cell stage, then it breaks down to 4-cell stage, and then the 8-cell stage, and moving on to the 16 to 32-cell stage where it is called a morula. Next comes the around 100-cell stage where it is called a blastula. Then comes the invaginating gastrula and the gastrula, where 3 different layers are formed, layers of cells which are called germinal layers: ectoderm, mesoderm and endoderm. I have discussed about this when I talked about the diploblastic and triploblastic organisms. So these 3 germ layers are formed, then it is called a gastrula. Then the blastula becomes a gastrula. So in all these divisions, if you can see that the cell number is increasing but the cell size is not increasing, therefore the cytoplasm content gets decreasing and the nucleus content is increasing. So this kind of division is known as cleavage, which happens whenever a single cell gives rise to a multicellular organism. So based on the amount of yolk present in the egg, types of cleavage can be decided. So yolk amount can vary from egg to egg, from species to species. Whenever there is absence of yolk, then we call it alecithal. Like in our case, we do not have yolk in the egg because our nutrition comes directly from the mother's body through the placenta, and we do not need an yolk for providing the nutrition. So alecithal eggs are those where yolk is little or none. And then comes the mesolecithal, where moderate amount of yolk is there. Then comes the telolecithal, where only at one end the yolk is there and the other portion of the egg does not have any yolk. Isolecithal, where the yolk is evenly distributed throughout the egg. Centrolecithal, where it is only present at the center of the egg and not outside. So depending on these contents of the yolk and distribution of the yolk, the type cleavage can be divided into the following types. So if the cleavage plane or the division is complete throughout the first cell, when the plane of division runs entirely through the first cell and divides the entire egg into 2 different cells, then we call it a complete cleavage or holoblastic cleavage. Like you can understand that this kind of cleavage is only possible when there is minimum amount of yolk, because since yolk is a very dense material, if a dense substance is there, then if we try to break a cell into 2 parts, then it will not allow the cell to completely give rise to 2 different cells. The cell division will stop here at this part and then there will be 2 different cells formed. But if there is no yolk at all, then the cell division will be complete and therefore 2 different cells will be formed. So this kind of cleavage is known as holoblastic or complete cleavage. But whenever there is a presence of yolk, as you can see, only the cellular part gets divided and the yolk part remains unaffected, and only it provides nutrition to the cells. So therefore this type of cleavage is called as incomplete cleavage or meroblastic cleavage. And according to the planes of division, there can be different planes. This is the equatorial plane. Just like in geography we have learned about the equator and the latitudes and longitudes, same goes here also. There is the equator, and parallel to the equatorial plane, these planes are called latitudinal planes or horizontal or transverse planes. And then one will be from the center, which is the meridional plane, and the other planes parallel to the meridional plane, these will be called as vertical or longitudinal plane. And Similar to that, there are two poles in an egg. One where the yolk content is maximum, that pole is called the vegetal pole, and the opposite pole where cell division takes place, this is the animal pole. Suppose this is the yolk here, so this pole will be vegetal pole and this pole will be the animal pole. So from that, according to the types of egg, the cleavage type can be determined, and you can understand that if it's little yolk or no yolk at all, then it will be holoblastic. If it is evenly distributed, then also it will be holoblastic. If it is mesolecithal or moderate amount of yolk, then it will be also holoblastic. But when it is telolecithal or an entire end is covered with yolk, then it will be meroblastic. Also in centrolecithal, the cleavage type will be a different, called superficial, because the central portion will be filled with yolk and only the surface will have the cells to divide. So this kind of special cleavage can be seen in insects. So there are examples also given here. Now coming to the last part, which is the tree germ layer formation. Like I said, that this is a blastula and the cells start invaginating or coming inside, forming a tube-like structure like this, and then they form three different layers of cells which are known as ectoderm, endoderm and mesoderm, and these three layers give rise to all the different organs and tissues of the body. So they are called germinal layers or the germ layers. And accordingly, this pore from where the cells start invaginating, this pore is called the blastopore. And whether this blastopore will form the mouth or it will form the anus in a growing organism, that decides whether this will be a protostome or a deuterostome. So in protostomes, in the example mollusks, annelids, arthropods, the cleavage is spiral, and in deuterostomes the cleavage is radial. In coelom formation is just like I have discussed here, so it will be like this, and the blastopore will give rise to the mouth. But in case of deuterostomes, the blastopore will give rise to the anus. So this kind of difference is there according to their fate, according to the fate of blastopore and coelom formation. So accordingly they are categorized as protostomes and deuterostomes. So here also, if you see that when it is an isolecithal egg that is evenly distributed, then it will be a radial cleavage, and there will be a spiral cleavage just like that. When the axis is spiral, that one cell will divide here, the other cell will divide here, the other cell here, the other cell here, so it will form a structure like this. But radial is that one plane will go just like here, and it will cut through the radius of the cells, and accordingly the cells will be formed like this. One plane will go from here, the other plane will cut from here, and so the one plane will be longitudinal, the other plane will be latitudinal, and just like that the cells will be formed. All cells will be equal. Bilateral is when they are dividing sidewise. So one like here, the other will divide like in the side, and then it will form a structure like this. Rotational is when it is rotating and dividing. When it is rotating and dividing, so forming an irregular structure like this. So the examples are also given here. And in case of mesolecithal amphibians, like I said, that this kind of cleavage will give rise to unequal cells. So they are known as micromeres and macromeres. There are two different kinds of cells in the blastula. Telolecithal, when the yolk is situated at one end of the egg, then most of the cell has the yolk. So therefore this kind of pattern will be seen, and only the few region or very little region of the egg will be forming cells. And in fishes, reptiles or birds where the yolk amount is very high, they will have a discoidal cleavage. So it will be a blastodisc, not a blastula. So a disc-like pattern will be shown by the cells, and therefore it will give rise to a blastodisc. Next is the central acythal, and like I have already said that it is called the superficial cleavage because the central part of the cell is the yolk, and therefore around this yolk there will be multiple nuclei formation and the cells will get arranged superficially around the yolk. So this kind of cleavage is called superficial cleavage. It is seen mostly in insects. So that is all for this part, and next thing is that we will start with pollution and conservation in the next video. So thank you.